Hello students, welcome back to the channel. In this video, we are going to study about oogenesis. So what is oogenesis? It is a process of the formation of mature female gamete and it is different from spermatogenesis. How? Why we say it is different? It is different why? Because it this process will start during the embryonic development only. When the oogonia are made within each fetal ovary, nearly millions are made within each fetal ovary, and no more oogonia are formed or added after birth. Means after birth, there is no more oogonia formation is done. So first difference is here only. In case of spermatogenesis, they will keep on dividing and increasing their number from puberty till the entire their reproductive life. Because in male, the spermatogenesis will start at puberty, where spermatogonia will go for mitosis to increase their number. But in case of female, oogenesis will start in the embryonic life and no more oogonia will be added after birth. Whatever they have to make, they will make in the fetal ovary. Then these cells will start division also, and now these cells after division, they are known as primary oocyte. They will enter into the meiosis 1 and they will stop at the diplotin stage. At the diplotin stage, they will stop. Means if you see the primary oocyte, whatever they have to be made is made in the embryonic condition only, and it has started the meiosis 1 and reached to the diplotin stage. Now, if we see the diagram carefully, we are finding that primary oocyte, this is primary oocyte, ploidy of primary oocyte is 2n. It is surrounded by few layer of granulosa cell. It is surrounded by few layer of granulosa cell. These are granulosa cell and this structure is known as primary follicle. This is primary follicle. So each primary oocyte is surrounded by a layer of granulosa cell and this structure is now known as primary follicle. And these follicles will degenerate from birth to puberty. It means at puberty only 60,000 to 80,000 primary follicles are left in each ovary. Is it clear? This much is only left in each ovary. At puberty, then each primary oocyte will enter into the next stage to make secondary follicle and tertiary follicle. How? See, understand, this is primary oocyte. Okay. You know very well, ploidy of primary oocyte will be diploid. It is surrounded by more layer of granulosa. Not only one layer, let me draw. Here we can draw 2-3 layers of the granulosa cells. Okay, these many granulosa cells and new theca. One theca layer is also made. So I'm just making it this way, you can understand. This is one theca layer is made. So this is theca layer and this is granulosa. And now this structure is known as secondary follicle. So I hope so primary follicle and secondary follicle structure is clear. Primary follicle surrounded by more layer of granulosa plus nutheca is known as secondary follicle. The primary follicle and secondary follicle, this much labeling might be easy for you. Now see next. Secondary follicle will change into tertiary follicle. This is your primary oocyte. Clear? And this is your granulosa. We are drawing it this way. This cavity is appearing. This cavity is fluid-filled cavity, also known as antrum. This cavity has appeared, and you are finding that theca is differentiated as two layers. This is theca interna and this is theca externa. So theca is differentiated as theca externa, theca interna. This was your primary oocyte. Labeling might be easy for you. And this cavity which is made, this cavity, okay, this cavity is known as antrum. And now this structure with all these things is known as tertiary follicle. This is tertiary follicle. And you know very well this is granulosa. So see the theory carefully. Secondary follicle will change into tertiary follicle, and tertiary follicle is the one which is characterized by the fluid-filled cavity called antrum. Theca will be organized as two layers, that is inner interna and outer externa. And now it is very important to see at this stage, it is very, very, very important line of NCRT. It is important to draw your attention that it is at this stage, means in tertiary follicle, primary oocyte will complete its meiosis division, which is unequal, making two cell, one large haploid secondary oocyte and first tiny polar body. So let me draw the diagram to make your concept easy. This is now secondary oocyte. And what is a ploidy of secondary oocyte is now haploid. This is first polar body. Okay. This is first polar body. And now secondary oocyte will secrete one layer that is known as zona pellucida. So see, this is the layer which is made by them. This is zona pellucida. The secondary oocyte is making this layer that is known as zona pellucida, which is glycoproteinaceous layer, or you can say non-cellular layer. Please learn it. It is not a cell, it is only a glycoproteinaceous layer. Zona pellucida, which is secreted by secondary oocyte. Clear? Then obviously now you can do the remaining labeling. This is granulosa cell, and these granulosa cells which are in touch with the zona pellucida, now they will be known as corona radiata. We will call them as corona radiata cells. This is the theca externa. First I'm drawing theca interna. You can see this is theca interna and then theca externa. So in this, you have to remember few things very carefully. Please understand it carefully. This is antrum. Okay. This is antrum. Please understand carefully. This is your secondary oocyte. Once again, I'm trying to show the labeling. This is first polar body. Clear? First polar body. This is blue color is zona pellucida, antrum, and let me just highlight with this color just for understanding. This is your corona radiata. Zona pellucida cells which are in contact, they are now known as corona radiata. Corona radiata. And this structure is now known as Graafian follicle. So this mature follicle is also known as Graafian follicle. I hope so this diagram labeling might be easy for you, and I've drawn step by using color difference to make you learn. The secondary oocyte will retain the bulk of the nutrient-rich cytoplasm. Tertiary follicle will change into mature follicle or Graafian follicle. And I told you very important line, the secondary oocyte will make a new membrane called zona pellucida. Is it clear? I hope so this much topic might be easy for you, and you have today learned all the follicles. Okay, let me just show you the outlook so that you can get an idea. See here, this was primary follicle with primary oocyte plus granulosa. Secondary follicle means primary oocyte more granulosa plus theca. And see, tertiary follicle means cavity plus theca externa interna, and Graafian follicle is where secondary oocyte, first polar body, and zona pellucida layer is made. See the summary. Primary follicle during puberty, this many are present. Secondary follicle, okay, which is having primary oocyte. Mature follicle will have secondary oocyte. Then ovulation will be done. After that, Graafian follicle will rupture, ovulation will be done. If fertilization is done, then only the second meiosis will be completed. If fertilization is not there, secondary meiosis will not be completed. So I hope so this video might be helpful for you. This video might be helpful for you. Thanks for watching.